Adams State College. Great stories begin here.
All right, we'll go ahead and get started, folks. Thanks for coming today. We do have a special group of speakers today. We don't normally have a collection of students, but we thought it would be appropriate. The Porter Scholars Program, which many of you are probably familiar with, was made possible through a gift by Mr. Porter, who this building is named after. And every year, students who are Porter Scholars are eligible to submit proposals for what we call focused academic programs. So these are programs beyond just the traditional scholarship where they're taking required classes and things of that nature. So some of these students were on the first year experience program that happens every May, that is put on as part of the Porter Scholars Program. Some of these students were doing some research over the summer, field geology and things of that nature. So, without further ado, and I guess I should also say that Dr. Jones and Dr. Vance were the individuals who helped put on the May session of the first year experience program for the Porter Scholars. Every year we try to rotate that through different faculty as well. So, having said that, I'll introduce the 5 students and let them introduce themselves as they go along. So.
Drumm.
Gary Potter.
Crystal Rodabaugh. Madeline, Brinkley.
Lassek.
And we did the Porter Scholars course over the summer, and what we did is we did the roller coasters and the international laboratories. So it was a week-long course from Monday to Friday, but on that Sunday we came in here and we got kind of used to the whole physics vibe of everything, and we watched a video on roller coasters, and we had some snacks like you guys are having today, and we kind of just got used to everyone being around. And then over the next 3 days, we had some courses on just the principles of different roller coaster aspects, and then we did some initial experiments. And we also had $60 to build our own roller coaster, as you can see here and here. We had 2 different teams. It was Gary and Crystal on one team, and then Madeline, Hill, and myself on another team. And we worked together during those 3 days to build our roller coasters. And then during those roller coasters, we had different aspects that we needed to achieve. We needed to have a hill, a free fall, a bank, and a loop. And then over the next 2 days after that, on Thursday and Friday, we went to 3 different laboratories, and also Elitch Gardens.
Okay, so some of the laws we studied before going about doing our roller coasters were the conservation of energy, the idea that energy cannot be created or destroyed, also including the idea that you can take potential energy and convert it into kinetic energy. Then we had Newton's 3 laws of motion: objects at rest tend to stay at rest, and objects in motion tend to stay in motion. Force equals mass times acceleration, and for every action there's an equal and opposite reaction.
To understand these laws, we have to learn a few definitions. And among them, you have potential energy and kinetic energy. And potential energy is basically the amount of energy stored in an object due to its position. So when you go up a roller coaster and it's pulling you up there, it's filling it full of potential energy, which then leads to kinetic energy, the amount of energy due to motion. And the cool thing is that the two ideally are supposed to be the same. You have all your potential energy, and then you start losing it, but then the kinetic energy picks up. And of course, unless there's friction involved, which complicates matters a little bit, and then that's still heat, or no energy is lost, it's dissipated as heat, but those are the 3 I wanted to cover just briefly.
And then our next one is, this is one of our initial experiments, testing the centrifugal acceleration. What we did is we had a bucket full of water, and we wanted to see how slow we can get it around our heads without spilling on each other. And luckily no one spilled. So. And then another one is one of the days we went to Carroll Park. And what we have is, Crystal's actually wearing an accelerometer, which looks like a 90s cell phone, which is about this big and this thick. And you stick it into a vest, and it tests, or it calculates the different points of acceleration that you have as you're in motion. And so we also took this to Elitch Gardens, and unfortunately, due to incompatible software, we weren't actually able to obtain any of the data points. Oh, and this, and these just are finishing touches on us working together and really trying to get our roller coaster done and actually working, which is a little challenging at some points.
This is the different parts of our roller coaster. We had a double loop, as you can see here. This is the nebulator, aka alien outlet. We had a bank right here, switch around the switchback where it fell down with free fall, hit the last loop, and stop at a little pipe loop. We also had to come up with a sales pitch for each of our roller coasters, and ours goes a little something like this. Explore the universe today on the alien, aka the nebulator. The double loop-de-loop passes through uncharted galaxies faster than the speed of light, with the alien light forms popping out at every bend. No one's sure of their intentions, so keep your distance. Try not to scream too loud on the free fall into what seems like another dimension. As you hit the final loop, it has time distorted and you begging for an outlet. With bright colors and innovative twists and turns, we'll have all onlookers lining up to have an experience that's out of this world.
Okay, so basically, we gathered information to compare theoretical and experimental calculations. The main ones I want to look at are g-force, right here. Calculated g-force is basically ignoring friction, and the approximate is taking in friction. And our calculations are off because we didn't have proper equipment. We used stopwatches to see at what point it took, to see how much time it took for the ball to transport through the roller coaster at different points in the roller coaster. So say we wanted to take the time from the top to the top of the sloop. We had to take the stopwatch at the top and then stop it in time for it to hit the loop, but that's not proper material to use to get those calculations, so they are going to be off. Like I said, calculated is ignoring friction and approximate is taking in friction.
Okay, so this is the other roller coaster we did, Loopy Larry. You guys actually fixed it. It was upside down last time I fixed that. So here's our first loop here. It comes down, goes around, and our first bank right here. I believe the free fall is right here, and then our curved bank. We ended up doing a corkscrew too, which wasn't part of the requirements, but we decided to do it anyway. And then right here we had an elliptical loop, and this was kind of interesting. It took a lot of time to figure out how to make it actually work without falling out. We also had a sales pitch, or it goes something like this. Loopy Larry has many exciting features that include a loop with a negative g-force, a tight lateral corkscrew, and a finale with the world's first elliptical loop. This ride is for the inexperienced riders still wanting to have the thrills of loops and curves. With a top speed of a whooping 13 miles an hour, your kids are sure to love Loopy Larry.
This is for our roller coaster. I had already explained g-force. The one before was for this roller coaster, but we only had one loop. We couldn't get the g-force of the elliptical loop because it's not a complete loop, it's an elliptical one. So.
And then this is our experience at English Gardens. And here is Hill with his helmet cam, and Crystal with the accelerometer on. And then we also, we got to play a couple games or whatever. And so this is Hill with he had a big hammer, and you have to try to smash it, and you have to get 150 points, whatever that means, in Carnival's top. And so if you play, then you get a little stuffed animal, and if you win 150, then you get a big animal. And this is me. I won the big animal. This is Hill.
She's stronger than both of us.
They won the small one, and my hammer's a little smaller.
One of the cool places we got to visit was Nist, the National Institute for Scientific Standards and Technology. And what's really neat is that somewhere off near Paris they have La Grande K. It's stored off in a vault. It's about the size of a plum made of platinum and iridium. And it's basically how they determine what a kilogram, or what a kilogram weighs. It's the standard. And then other people come bring theirs to that place, make sure they weigh properly. Lots of countries have these. The problem is that they're losing mass. from cleaning or whatever. Its weight is fluctuating, and that's not a good thing. So the solution that a physicist and a mathematician, Dr. Ronald Fox and Theodore Hill, they came up with an idea of something based on nature, where they take the mass of a lot of carbon-12 atoms, and they're going to use that to be the new standard of measurement if it gets approved. And we're talking about 18 times 14,074,481 squared carbon-12 atoms. So a lot of atoms make up a kilogram. And what's neat is their ideas based on nature. You have like length, time, the second, current, which is like an ampere, temp, which is Kelvin, and substance, a mole, luminous. All these are based on nature, but so far our weight system, our kilogram, is not. So that's what they're trying to do here. And what you see in this photograph on the right-hand side is an atomic clock, and that's in Boulder. They keep track of what a second is, which is, we kind of take that for granted, a second's a 1/1000 to 1,000. And they're actually working on something even better than that. And, Jason Whittegren, you see him right there in blue, he was our tour guide, really awesome guy if you're ever in Boulder.
Okay. Next, we visited National Lab in Boulder called NOAA, also known as the National Oceanic and Atmospheric Association. We were briefed on tasks the lab was doing in regards to current issues in the environmental world. The tour started in the Space Weather Prediction Center, which is in this picture. Through these glass windows, you can see actual pictures and images of the sun. They study the space weather's interaction with Earth's patterns. They examine things like plasma bombs and sunspots that could possibly interact with our Earth's magnetic field, which would help predict if we need to prepare for something like that. Then we were shown, in my opinion, the best part, then we were shown the center where they collected air samples from around the world, over 20,000 different locations around the world where they would measure the CO2 amounts in the air sample, and that's where they get their current fluctuation is actually 390 parts per million right now, and that's increasing at an exponential rate, caused by deforestation and also fuel burning, but that's another subject. Then we were shown, in my opinion, the most interesting part, which is this picture right here. This is science on a sphere. You walk in and there's nothing in the middle of the room, and then all of a sudden they push a button on the remote. And 4 separate projectors around the corners of the room project an elliptical image that together forms a ball that you can basically walk around and you can see all the different Earth patterns. They showed us things from volcanic activity, hurricanes, airplane routes, formation and degradation of continents, decreasing polar ice sheets, and more. So that was really interesting. And they have over 250 databases on that actual science hemisphere that you can see online. These are a great way to look back into the past, and perhaps the most amazing feat that we can see other planets on this sphere. Not just Earth, they could look at Venus, Saturn, Pluto, which is all a little fuzzy because of the distance. That's about it for the NOAA visit. This lab visit really displays the impressive scale of discovery human technology has reached.
Alright, so the last lab we went and visited was NREL, and that's the National Renewable Energy Laboratory. The mission and strategy is focused on advancing the U.S. Department of Energy's and our nation's energy goals. The research spans from understanding renewable resources for energy to the possibilities of these resources, to renewable energy, electricity, and fuels for the homes, commercial buildings, and vehicles. So therefore, this laboratory directly contributes to our nation's goal to finding new renewable ways to power our homes, businesses, and cars. The types of alternative resources include wind and solar, so you can see these are solar paneling right here. And they do biofuels and geothermal as well. Video.
technical difficulties. So we'd like to conclude our presentation.
If you would like to ride the nebulator in the audience, city volunteers? Oh, sure.
Be careful, it's dangerous.
All right. Should get stuck right about there. I'm gonna get stuck.
Oh, man.
That's it.
Anybody have any questions?
I want to know where it was, on the nebulator, where it was faster than the speed of light.
Well, you can't really see it.
Actually, I was wondering, you mentioned about the carbon atoms for the kilogram.
Yes,
How do they count those?
that's a good question. They have, it's a two-story mechanism that, I don't know the details behind it. We weren't even allowed to see it. It's like top secret or something. That's a good question. I don't know.
So while the nebulator was being created, I had the opportunity to attend Geology 495, which is field camp for geology majors. It is our version of the same capstone. It's 6 credit hours. We cover it in about 4 weeks. Monday after graduation, we were driving to New Mexico. But a little bit more about field camp first. All right, so what is field camp? According to the course catalog, it is supposed to be a course designed to allow students to learn how to use traditional geologic field mapping techniques. According to the students that had to do it, it involved a lot of hiking on rocks and a lot of hammering of those rocks. So, oh, there it goes. Okay, so in order to do that hiking and hammering, I'm going to tell you about 2 locations. We visited about 5, but there are people in this room who have not yet had the opportunity to take field camp, so I'm not going to ruin it for them. We started out in Alamosa, there's our X. For our first section of the trip, we went down to Brinkley, New Mexico, which is just a little bit north of Albuquerque, and we spent some time at a place called Tenorio Ranch and White Mesa. We next went up north into Colorado, and we were outside of Salida. We were in Buena Vista and Nathrop and Wellsville. I will be talking about Wellsville. These are a few pictures from Tenorio Ranch. It's actually a beautiful place. It looks kind of desolate in these pictures. I'm sorry about that. The problem with geologists is that we don't take a lot of pictures with people in them. We tend to take them of landscapes and of rocks and with people for scale. So we were on Zia Pueblo property for a little bit of this. The Zia Reservation is actually located next to an area called White Mesa. It's called White Mesa because it's made primarily of gypsum, and we're mining it industrially. I'm trying to get that to pass around in a minute. This exciting feature here is actually part of the White Mesa complex we were studying, and we were actually on top of this. We climbed up the top and mapped all the way back. It's about a 1/4 mile long. Prior to that, we were over here back in this area along this access road. We weren't allowed to go on this side. That was exciting too. If you transport yourself over to the tribal properties, they get the site. So, but here we were looking at sedimentary structures. You can see there's a lot of layering going on. The sneaking suspicion there is that there's different rock types in these layers, and sure enough, we confirmed that. So, after that, we went to Wellsville. It is on the way to Canyon City if you're leaving through Salida. And we got to deal a lot more with some sedimentary stuff and with some igneous basement rock as well. It was very, very old. The here's a view looking out over the valley mapping, and we actually started down here and worked our way up, and then worked our way up and around. This picture is a little bit further up. You can see the person for scale. We didn't actually make it into this cave. We decided that wasn't a good idea. We ended up going out further to the top. And here, this is special for all of you that haven't taken field camp yet. Sneak peek of the private area where we went for our final. So, there you go. What did we do at field camp? We hiked. This is again at Tenorio Ranch. This is shale. This is very nasty. Don't climb it after it's rained. We did a lot of adventuring. We were supposed to be doing field mapping. The field mapping component means that you're out and about and you're actually walking on this stuff. You're not just looking at it in a picture. You're not just guesstimating from afar what it really is. You find a contact between two different rock units and you walk it. So we were walking the contact. In this particular case, we were measuring what we call a stratigraphic section. The strat section is a layer cake. Ideally, it's a listing of all of the units in an area, the different types of rock, and how they're related to each other. It's looks like a neat stack. Preferably you have the oldest stuff at the bottom. Sometimes it doesn't work out that way. But that's what we're doing here in our puzzlement. We have just dropped a rock on the ground that marks 0 for the stratigraphic column, and from there we're measuring with these things called Jake staffs. These are a 5-foot pole with a 10-foot increment all the way up. And we use those when we're documenting the thickness of those units that we're tramping all over. As we were doing that, we were also taking observations at a closer scale. These things are called Chert Nodules. We found these in one of the units not too far from where we were standing here. Things like this, these close observations can give you a lot of information about the environment, and that's what we were out there to do. We weren't just looking at what's where, we were looking at how it got there. So further up, we saw things like this. This is a chert unit as well, but it looks very, very different. And these are what we suspect are some sort of water features. So the field book is in there for scale, F-scale. I have some samples of what we looked at there. That really nifty chert stuff down in this corner. These around. They're actually quite lovely if you can see them. I might turn the lights on. We also do climbing in Wellsville, as you can see. I believe the retrieval point for those rocks that I just started passing out was somewhere around here. There was also a timber rattler somewhere around there. So we got a little bit of a biology lesson. That was exciting. Herpetology was incorporated in the sense that you wanted to avoid the herps. And the last cool thing that we got to do, this was kind of a bonus that we weren't expecting. We were planning on being dragged out for a horrible, horrible day of mapping in the heat and sun. It was going to be awful. And we ended up riding the train. We took the train from Antonido down to Charm and back. And while we were out there, we got to see some absolutely beautiful features, including a lot of great examples of glacial geomorphology. This is the Los Pinos River running down through a beautiful U-shaped valley that was created by a glacier some time ago. We enjoyed the views quite a lot, and a lot of my friends didn't realize that I had a camera. That was funny. They turned around and took a picture. But when we weren't doing that, we were actually there to further apply what we had learned, since the whole point of field camp is to use all of the knowledge that you have acquired thus far in your geologic student career and learn more in the process. We were allowed to teach the docents on the railroad line a little bit more about what they were seeing, because these gentlemen are out there, the ones in the hats, let's see, there's 1, 2, 3, 4 of them right there. The gentlemen in the hats go on these trains on a regular basis, and they are required to tell other people about what they're seeing. So we told them what they were seeing so that they could share that information with others, and they pushed us pretty hard. It was pretty brutal. But overall, it was very enjoyable, and I think by the end of our final field mapping experience, we were all feeling pretty good about ourselves. We got there and thought that we were not going to know what we were doing. We were all thinking, oh my gosh, this is going to be absolutely terrifying. I don't think I can do this. And by the time we left, we felt pretty good. So I will leave you with this quote. This is advice from Charles Darwin to Charles Lyell. Make them, like me, adorers of the good science of rock breaking. We, by the end of field camp, all of us were that hadn't been before.
While they were working on roller coasters,
And they were working on rocks, we were working with DNA. So, quite a variety of different things that were going on this summer. Josh, along with myself and Dr. Bedard, started working on what we now call Molecular Biology 2 here at Adams State. So we're going to give you a brief overview of what we were looking at. The first part included me going to St. Louis. St. Louis is the head of the GEP project, Genomics Educational Partnership. And this is just a map showing all of the schools that are currently involved in this project. Well, we are the first school in Colorado. This is Highlands, New Mexico, for all those who are interested. But besides California, there's only 3 other schools kind of on the western half of the United States that are involved in this project, and so I definitely feel lucky that we are included in those schools. This is what I saw when I first went there, and I felt like I was walking into a castle. So this is Washington U. This is the Genomics Center at St. Louis. This is where all of their sequencing machines are and where all of their lab work is done. So the thing that we were going to be looking at, I thought I was done with flies at fall semester, but I was wrong. So at the end of last fall semester. So the species that we were looking at were D. mohabensis, D. grimshaw, D. erecta, and D. melanogaster is the comparison species. Now, for the genetic DNA that we were looking at, this species has already been finished and annotated, which you will soon understand what that means. But we are in the process of doing an annotation and finishing for erecta, mohabensis, and grimshaw. Okay, so Josh is going to tell you about the wet lab part.
Okay, so part of our focus was concentrated on a process called finishing. Now the Genomics Center that Amber had a slide of a while ago, in that center they sequence DNA. So there are adenine, guanine, cysteine, and thymine that all code for proteins in DNA. And what the sequencing center does is say what order they're all in. But sometimes there are sequences that you get one nucleotide that repeats too many times, and the machine has a problem reading it. And the machine goes so fast that it just skips over them. So an analogy would be you're reading a book and all of a sudden you get to this chapter where you're missing a paragraph or a whole paragraph doesn't make sense. So what does it mean? Why is it there? And our purpose is to clean that sequence up so that other scientists can use that sequence and figure out what's going on. So here's some of the equipment that we used, a workspace, and some of our reagents. You do not believe how expensive a small amount of taq polymerase is. $40 for less than a milliliter.
Yeah.
So some of these reagents have to be stored in a freezer because they're temperature sensitive, and if you leave them out in room temperature for too long, they will not work as efficiently or at all. The solution was sent and left at room temperature for a weekend, and it didn't work at all, so we had to get another one. So, and all this material right here was all provided to us by Porter Scholars. So the basic idea that we have to start out with is we have a bacterial cell. It contains its own DNA. It contains these pieces of DNA called plasmids. And scientists can take a plasmid and insert sequences of DNA that we want. So what they'll do is insert the sequence of DNA that we want to finish, and they'll give it to us in a petri dish. Each of these colonies is a colony of E. coli with a plasmid in it. And as the cell replicates, it continues to copy this plasmid. So each time it replicates, you get you double your amount of plasmid. So we let the bacteria do most of the work for us. And what we also had to do was make a cryogenic vial, glycerol stock. And that was because if this ever got contaminated or went bad, we would then have a sample of bacteria to continue working with. So the next thing we had to do was we had to grow the bacteria, and then we had to run a plasmid prep to isolate the primer from the rest of the bacterial contents. And at the end of that process, what we had was a solution completely made of primer. Completely made of plasmid, excuse me. So the next thing we had to do was develop primers, and what they are pieces of DNA that will bind to the plasmid, and you can imagine them like bookmarks. So you mark one end of the DNA, you mark the other end, and then that whole section in the middle is copied by a protein, and you do it in this machine, and it does it 1,000,000 times over, so you have a very high concentration of plasmid, and specifically the sequence that's in the middle, which is the sequence that we want to finish and resequence. And just for a size comparison, this spot right here is this slide, or this picture, and we have a nickel. Our tube is barely larger than the diameter of a nickel, and we only use maybe about that much as our reaction volume. So we're dealing with very, very small amounts in each tube. So the next thing we want to do after we run the PCR and we have copies of our plasmid sequence, we want to make sure that it's actually there. So what we do is run gel electrophoresis, and what we have here is our power source, and we'll put the gel on here with the DNA in these wells, apply electric current, and the DNA will run towards this positive electrode. And smaller pieces of DNA will run farther because they're able to move through the pores of our agarose gel. So what we're seeing here is a DNA ladder, and these are is part of the is a chemical that we had to purchase, and it contains certain sizes of DNA that are known. And they give you an illustration that shows you what each of these bands represents as far as how long it is. So we see all these bands here are all the plasmid that we had, or the DNA sequence that we had isolated from the plasmid. And right down here, these faint parts right here are all meaning that we still have the primers, because they're extremely short and they don't even show up on the band of our ladder that's below us. And then in this last lane, you notice that there is no plasmid sequence right here, and that was intentionally done because we wanted to do a negative control. So if we had any contaminant DNA that was in there that the primers could bind to, we would end up with a band similar here. But since we have none, we know that we had a good reaction mixture with only our plasmid sequence. So the next thing we want to do before we take it to the sequence machine is purify it. And to do that, we just want to remove the primers so that all that's left is our sequence that we took out from the plasmid. And then whenever we do that, or we finish that technique, we do another gel. And we have our ladder here again. We see two bands for our bacteria, or for our plasmid DNA sequence, which is in the same position as right here. And we have absolutely no bands down here where we would expect them if we had failed to do a good purification. So we know now that we have only the plasmid that we want to be sequenced. So what we do is we sent it to Cincinnati University. They ran it through their sequencing machine, and what we would do is get a readout. This right here is a readout of one of our actual projects. And this is the kind of stuff that is called pore quality sequence. So all these peaks represent a nucleotide base, and then they're red up here. As you can see, they're overlapping. Some are very broad. It's very messy and very hard to read. And what we want to accomplish with these techniques is to be able to convert this to something like this. Very good, high defined peaks, nothing overlapping. And so with our plasmid, we just used a piece of the plasmid that we had already known the sequence to. Took it to the sequencing in Cincinnati and got a readout back, and we were able to successfully isolate a section of DNA from the plasmid and have the correct sequence. So we now know that anytime we need to isolate a sequence of DNA, that we are fully capable of doing so.
Okay, and so after that, we do the computer work. And so, just for those of you who kind of need some of this basic information, genes are what make us who we are. They help what color of hair you have, what color your eyes are, how tall you are, things of that sort. And so what we're going to be looking at are the components that make up this gene. And these are called exons. So this is what we're going to be looking at on the computer when we get that finishing result back. So the first thing that you're going to see here that I want to point out is this sequence up here. Now this sequence is the sequence that went through all of the wet lab procedure and we fixed it, and so now we know what is a T, what is an A, what is a G, what is a C. And this sequence down here, if you remember previously, I told you that we know D. melanogaster, which is that one fly that was at the top. We know its sequence. And so what we're going to be doing is comparing D. erecta's sequence to melanogasters to see how similar they are and get a general idea of what it's going to look like. Okay, so when we go into that program that is called the genome browser, we look at we look for certain spots in the sequence that line up with, that correspond to the start of an exon or of a gene and the end of an exon. So at the very beginning of a gene, we have a sequence that codes for a methionine amino acid, and that is the ATG site. So that would correspond to a sequence right here, if this is the beginning of our gene. So we look for that. Then we look for a GT site, which would be the end of the first exon and every subsequent exon after that. And then we look for a G sites, which are the starts of the exons after the first exon in the gene. And so we look for these sites and try to find what nucleotide number they correspond to. So if I'm looking at this A right here, it corresponds to number 37,455, the 55th nucleotide in the sequence that we looked at. And so there's a lot of nucleotides that we're examining here. And so what we do is we write down these nucleotide numbers. That way we know where the start of the exons are and where the end of the exons are. And there's a whole lot of other complicated things that go on, but that's okay. We'll just skip that. And so, poof, with magic, we come up with this sequence right here. A lot of hard work, actually. But and so what we're looking at is the sequence that we processed in the wet lab, and then we went ahead and annotated it. And this right here is our sequence from the wet lab, and this is our sequence that we already knew, which was D. melanogaster sequence. So this is that other fly, D. erecta, and this is melanogaster sequence. And what we can see here is that many of their exons, this is the entire gene here, and these are all the exons that are in the gene. And what we see is that there is a lot of similarity between the two species, but there's also differences. Especially a good place to note is that this end exon right here continues on, while in melanogaster it is broken into two separate exons. So this is kind of what we're looking at. So we take all that information, we plug it into this computer program, and basically what it does for us is it checks to make sure that all of our information corresponds to the actual numbers that are in the sequence. Say we may have messed up a number here, it would say fail here. But this is a pass, so all of our sequence numbers are good. So we take that and we send it to St. Louis, and they put it in their file, and so then they can use that information for further research on the fly itself. So, and it's used for multiple avenues of research. But that is kind of basically what Josh and I did this summer. Are there any questions? Oh. I would like to note that we are doing another talk in November, another lunchtime talk, and we're going to go into a lot more detail
kind of showing you exactly how the process of annotation works and kind of exactly a little bit more information on exactly what we were looking at and how we did what we did. So come to that lunchtime talk, please.
Adams State College. Great stories begin here.